synaptic vesicles are going to migrate towards the surface. This is all. Remember, this is a three-dimensional structure, okay? So that axon terminus is sitting there kind of like this, sitting in the synapse. These vesicles are produced further up, and they contain neurotransmitter. They are going to merge with the plasma membrane, and the neurotransmitter is going to be released into the synaptic cleft. The synaptic cleft is the area here between the presynaptic neuron and the target. Are we all still okay? This is a great, you might be a little removed from all of this, and it's a great time for you to ask questions. So stop me anywhere along the way here and say, Kel, wait a minute, I'm a little bit lost. Okay? Now we have neurotransmitter in the synaptic cleft. The synaptic cleft is everything around your cells in your body has fluid. There are no dry cells in you. The only dry cells you have are on the surface of your body. So we've used a change in the membrane potential to bring about the release of a neurotransmitter, which is going to carry that signal across the cleft to the postsynaptic membrane, where we are going to have gated channels again. But the neurotransmitter binds with these channel proteins, and when it binds, they open. These are ligand gated. So the neurotransmitter chemically acts as a ligand. If you know chemistry, that makes sense to you. If you don't know chemistry, just go with it. Okay? It's the key that opens those channels. The channels that are within the synapse are what we call biports. Ions can move either way. We start to see sodium ions entering the target. We start to see potassium ions leaving. And we start a graded potential. Now, here's where all sorts of additional interesting things happen within the synapse. The neurotransmitter that we've released here will bind to these, and as long as it's bound, those channels are open. Some of the neurotransmitter will leak out. That neurotransmitter will be picked up by astrocytes and returned to the axons. So it's not wasted. Some of the neurotransmitter will be taken up by reuptake. So while the axon is releasing neurotransmitters, some of it goes right back into the axon. And some of it ultimately will be degraded. And degradation has to happen. If you do not degrade the neurotransmitter, it's going to stay bound to the receptors, and those receptors will stay open. And as long as those receptors are open, you will just keep generating action potentials. So this is important to understand, because this is how signals move everywhere in your nervous system. We go from voltages, millivoltages, changes on the surface of neurons to neurotransmitters in the synaptic clefts. Now, so here is that graded potential. Here's the integral channel protein. Here's the plasma membrane, the neurolemma. Ions are moving in both directions. We have the graded channel. As that moves away from the synapse, we are going to approach voltage-gated channels again. So everything we've looked at up until this point is happening here. Once we get away from the synapse, we have voltage-gated sodium channels. During depolarization, sodium is crossing the membrane from the outside of the cell to the inside of the cell. Sodium is positively charged. As we bring more sodium into the cell, the inner surface of the membrane becomes more positively charged. That is the depolarization event of the action potential. Are we okay still? Yeah.
Okay, so with graded potential, the calcium channels are open.
Yep.
Correct, and then during the action potential, the sodium is crossing into the membrane. I'm just making sure I have it correct.
No, yeah. So in the target, okay, so the calcium channel, the calcium channels that are open are in the axon, okay, the axon terminus. Once the signal gets through there and you release your neurotransmitter, those can actually start closing. Okay, it's like the job has been done. We've moved the vesicles, we've released neurotransmitter. Now the neurotransmitter is binding to the target in the postsynaptic membrane. That's where the ligand-gated channels are opening, ions are moving both ways. But the signal is going to diffuse away from the synapse along the surface of the target. Once you have left the synapse, now you bump into voltage-gated channels in the target, on the postsynaptic membrane. Does that clarify that a little bit? Okay. So we start sodium rushing in. As sodium rushes into the cell, the membrane potential changes rapidly. We go from -55 to +30 in about that amount of time. As you approach +30, two things happen. Number one, the voltage-gated sodium channels close. But potassium channels that are voltage-gated open. Potassium normally is on the inside of a cell. Potassium, just like sodium, is positively charged. So here we're moving sodium ions, here we're moving potassium ions. And potassium now starts to leave the cell from the inside to the external parts of surrounding the cell. We cross the threshold, we keep going, we cross the resting potential, we keep going. Once we cross the resting potential, we are now hyperpolarized. Remember, we're at about -75 here. That's beyond where you want to be. Why do you think we go beyond where you want to be rather than just going right to where you need to go?
Recharge, or not recharge, but
However, make sure.
I'm trying to think of the right word of recharge, but it's
Yeah,
basically.
we are bringing, we're kind of, remember, we depolarize, we're repolarizing, so we are kind of recharging, we're taking the charge back to where it is. However, what were you about to say?
But you say like it goes there so that way here's no more just like make sure because the K channels are still open at that point.
Oh, these are all great ideas.
Actually, I think it's because, if I can say that, it can't go backwards, pretty much, it'll keep going,
Functionally, that's phenomenal.
but no.
No, you all have a piece of the puzzle, okay? The reality is, these cells are stupid. They can't count. Now remember, to get the charge right where we need to go, we have to move the exact number of ions across the membrane. They can't count. So you overshoot the target to guarantee that you're going to be back at -70. Because all cells in your body have a very important pumping mechanism that I find students try to erase from their memories. And that pumping mechanism is the sodium-potassium pump. The sodium-potassium pump in cells is always working at low speed, okay? To guarantee that sodium is on the outside and potassium is on the inside. Which is why this takes a little while to get back up to -70. So the reality is, it's guaranteeing you're going to have this reaction. It prevents it from backsliding, but at the same time, it guarantees that you're going to get to -70 again. But it overshoots it. Because all chemical reactions that take place in you, regardless of whether it's your nervous system, your digestive system, whatever, you produce more than you need. Because if you're cheap, which our bodies are and our cells, you want to produce just enough. All right? But if you produce a little bit less than just enough, what happens? Nothing. Worst case scenario, cell dies. Oh, that's pretty worse. Yes. So by overshooting, going into the hyperpolarized area, it guarantees that you're going to get to membrane potential without needing to count. Okay? So, yeah. Now, you send a signal. But in reality, when you send a signal, you know, we're going to do an exercise. Okay? Take your hand, put up fingers, take your other hand, wrap it around a couple fingers. Just in contact, right? Now squeeze. Squeeze. Squeeze till your fingertips turn purple. All right? Okay, I'm teasing. What's the difference between that? When I was just in contact versus squeezing. Both of those and everything in between, was I using the same set of muscles? Yeah, I was. And all of what was happening there was degree. Oh. Okay. So, let's try something else. Lift your hand up. Put it down. Lift your hand up. Put it down. Lift your hand up. Put it down. How can you do that? All right, you can stop. Lift your hand up means you have to get a series of action potentials to do this. But now you stop and put it down. Are you still getting those action potentials? No, you're not. Well, how did you stop them?